Natural products are often biosynthesized as a class of compounds and not as a single product. Diagnostic fragmentation filtering is a simple approach for detecting all structurally related compounds within a complex mixture.
Diagnostic fragmentation filtering, implemented within the MZmine software, allows the analyst to explore mass spectrometry datasets for new natural products that would otherwise be missed. This includes new bioproducts and also new toxins.
Diagnostic fragmentation filtering applied to toxin classes that contaminate food and water can inform exposure assessments. When applied to microbial and plant extracts, it can result in the discovery of new compounds. Using diagnostic fragmentation filtering requires an understanding of the key tandem mass spectrometry features that define a chemical class. This means that structurally similar compounds will generate characteristic ions.
My name is Shawn Hoogstra. I'm a research technician and software developer at our center, and I will be demonstrating the procedure.
To prepare a sample for microcystin analysis, inoculate 30 milliliters of sterile cyanobacteria growth medium to approximately 5 × 10 to the 5th cells per milliliter under aseptic conditions in 250 milliliter Erlenmeyer flasks, monitoring the cell density with a hemocytometer. After 26 days of photoautotrophic culture, illuminated with cool white fluorescent light using a 12-hour light-dark regime at 27 degrees Celsius with swirling once daily, use 47 milliliter diameter GFC glass microfiber filter papers to separate the cells from the culture medium by vacuum filtration.